Good afternoon, everyone. Welcome to the H3D Foundation webinar series. Our today's speaker is Dr. Clint Veale. Dr. Veale is working as Associate Professor at the University of Cape Town. He has interests in organic chemistry, chemical biology, and biophysical chemistry, and its applications to drug discovery and medicinal chemistry. So today's topic for webinar is old targets, new tricks, protein-protein interactions, and the extracellular environment as new modes of HSP90 inhibition. Before we start the webinar, the general rules that we'll have a Q&A session after the talk. So you can either post your questions in the chat during the talk, or you can ask it at the end of the talk. Now I would like to welcome Dr. Veale, and over to you, Dr. Veale. Let's hear.
All right, thanks very much. And just to start, can you hear me okay? And can you see me all right? Or see my presentation okay, at least?
Yes.
Perfect. All right. Thank you very much for that. And as was indicated, I'm going to talk a little bit today about our work in the HSP90 space, and particularly trying to find new ways of exploiting this very promising target. And we'll get into more of that now. So to jump straight in, what we have here is a typical tumor environment. We have our lovely pink healthy cells, and we have our ugly kind of purple wavy cancer cells, and these together form what we would consider a tumor. Now, looking inside that environment, what we have is what we see is that what's called the HSP90 cycle, and this is responsible for taking proteins from the coming right off the ribosome in their native state and getting them into their folded and active conformation. So how this happens is HSP90. Yes.
Sorry for the interruption. There is a comment that participants are unable to see your slides. So can you please stop sharing and share it again?
Let me try this. Hold on a second. Sorry about that. Let me try it this way around. Can we see that? What can we see?
Yeah, I can see.
Can everyone else see? Can I see the actual slides or?
I can see your PowerPoint. Yeah, and slide number 6.
All right. So are we happy? Is any comment from the audience?
So yeah, it's not on the presentation mode yet, but I think now everyone can see.
It's not. Well, it should be on presentation mode. What about now?
Not yet.
Can you not see it?
I can only see slide number 6.
Oh, dear. What's happening here? Okay, sorry. This might take a bit of teething problems here. Q&A saying we can't see slides. Thanks, Vin. On top of it. Try. Is that in presentation mode now?
Yes.
And Vin, can you see it? I can't read the comments.
No. Yeah, people can see it now.
Hey, okay, happy days. Okay. So I don't have a pointer anymore, but that's okay. If you can see my mouse, that's useful. So what happens in this HSP90 cycle is that heat shock protein 90, which is this diamond orange, forms a series of protein-protein interactions with the co-chaperone hop and HSP70, and it facilitates getting nascent proteins into the active conformation. So this process is critical for cellular proteostasis in all cells, but particularly in the cancer environments when you consider the high cellular growth rates and the interface with mutated oncoproteins. Furthermore, several client proteins for HSP90 include signaling intermediates and transcription factors. So this target is seen as a very promising way to inhibit cancer growth and inhibit cancer biology. Now, due to that fact, there's been a lot of effort put into generating HSP90 inhibitors for cancer drug discovery, and here's 3 shown here, but there are many others. And whilst these compounds tend to perform quite well in the early phases of drug discovery, they have a tendency to fall out later on in the drug discovery pipeline, the early clinical trial phases. Now, one property they have is that they stimulate something called the heat shock response. Now, what this is in the presence of, when you inhibit HSP90, you get this compensatory action of HSP70, which gets overexpressed, and it can take some of the load away from HSP90 to preserve cellular proteostasis. So that's one hypothesis as to why HSP90 inhibition has been less successful in the later stages. Now, HSP90 also tends to have a quite prominent extracellular role. So it has been shown to interact with cell surface receptors, which and is involved in regulating cell signaling and, of course, cell migration, whilst HSP90 has also been shown to have an important role in remodeling of the extracellular matrix. So together, HSP90 also plays a critical role in cell migration and metastasis, and it's now hypothesized that possibly the reason that HSP90 inhibitors do so well early on is that extracellular HSP90 might be the real target for HSP90 inhibitors, and kind of common modes of drug discovery confound this because we tend to optimize for things to get into a cell. Right? Somewhat peculiarly, there's been no real effort to design compounds to particularly target the extracellular environment, however, to at least try to confirm this hypothesis. So, given the role HSP90 has in cancer biology and its obvious properties as a promising target, we started thinking, how can we rethink HSP90 inhibition to get around some of these pitfalls, whilst in order to try and, as I said, find something that can target HSP90 effectively. So the first thing was, can we modify molecules to limit cellular penetration and take advantage exclusively of the extracellular environment? And the second one, which is a lot more developed project, is can we inhibit the protein-protein interaction between HSP90 and its co-chaperone HOP, shown in green, and potentially perturb HSP90 function in the cell without stimulating the heat shock response. So I'm going to start with the extracellular HSP90 inhibition story, and this kind of started when we came across a paper by Walker et al., where they modified a series of luciferase inhibitors without disrupting the pharmacophore. They tethered on this alkyl sulfate chain, so this disordered, flexible polar group. And what they were able to do was alter the luciferase inhibitors into exclusively extracellular luciferase inhibitors. So to that end, we thought we'd work with these SNX compounds, and these are N-terminal ATPase inhibiting HSP90 inhibitors. We chose them for a number of reasons, one of which was we thought they'd be a little more synthetically tractable, but that was a whole story of its own, but that's not for today's chat. And we also selected these because there'd been a previous study where an SNX analog had been tethered to a fluorophore to try and determine cellular localization of HSP90. So we used this information to design 4 compounds. So we retained our SNX pharmacophore and we tethered to a phosphate or sulfate alkyl chain. And the lengths of these chains were really dependent on what was commercially available at the time. We also did some in silico stuff. There was fortuitously, there was a co-crystal structure of SNX2112 crystallized in with HSP90, and our compounds, when docked in, occupied a similar, in purple, showed a similar binding mode to the parent inhibitor, and our alkyl chain orientated outside the pocket, indicating that it wasn't going to disrupt target binding, which is of course essential. So the first bit of biological data we got for these compounds was cytotoxicity, and here's SNX2112, which is a potent cytotoxic agents, got a PRC50 of about 7.5. And our compounds lost about an order of magnitude, although still pretty potent. 6.5 is still in the mid-nanomolar range. And these compounds were found to be pretty good cytotoxic agents. The drop off, we're not sure why, possibly because we weren't engaging with the intracellular environment. An absolutely critical experiment was, of course, determining if these compounds were still able to inhibit HSP90, and we got some data to show that these compounds, this is normalized to GMSOs as the control, that compared to SNX2112, all four of our modified analogs retained the same, more or less the same ATPase activity against HSP90 as the parent compound. So that was a very important result for us because we were showing that we were still capable, at least, of inhibiting HSP90. So moving on to the intracellular environments, the first thing I wanted to look at was if we were stimulating the heat shock response. So we look at HSP70, and in the presence of a bona fide HSP90 inhibitor, we see an increase in the expression of HSP70 relative to the expression of the levels of the histone. So this is a typical HSP90 inhibitor, and as I mentioned earlier, we get this indication of the heat shock response. Importantly, our compound showed no such effect. We didn't see any increase in the expression of HSP70 in the presence of our putative extracellular inhibitors. We then looked for the impacts of these compounds on a HSP90 client, and we looked at CDK4. Now, of course, as an HSP90 client, inhibition of HSP90 causes the levels of this protein to degrade, and of course, SNX2112 showed a corresponding degradation of CDK4. Similarly, similar to the previous trend, our compounds didn't seem to have, apart from one data point here, didn't seem to have much of an effect on CDK4, almost looking to increase the levels somehow of CDK4. So these compounds indicating that this data indicating these compounds don't impact the HSP90 client. Excuse me. So together, our data showed that ATPase activity was retained, so our positional modification was suitable. Suitable cytotoxicity was retained. This is possibly due to extracellular activity, but that's not confirmed yet. HSP70 levels remain stable. HSP90 substrate was not degraded, which together are strong indications this compound was not entering the cellular environment. And of course, we don't know if this is going to mean anything yet, so we currently are looking at a matrix remodeling and wound healing assay, which is currently underway, and this will give us more of an indication whether exclusively targeting the extracellular environment has any translatable effects on cancer biology. So that's where that project is currently. Moving on to the protein-protein interaction space. Now, the first thing to look at is what kind of targets we use in drug discovery. Now, traditional drug discovery, we tend to look at very similar classes of targets. I'm talking about G protein coupled receptors, ion channels, enzymes, kinases take a lot of space. And this has been immensely successful in developing a lot of inhibitory compounds for drug discovery. An important thing to consider is that many of the biological roles of these targets are actually mediated by very specific and directional interactions between partner proteins. And it's these partner proteins which will activate many target proteins into their active conformation or get them ready to do their biological response. So the protein-protein interaction community see if we can disrupt these protein-protein interactions rather than directly targeting an enzyme or target, we are giving ourselves an additional opportunity to inhibit a biological response and biological pathway. Our recent study using a yeast-2 hybrid system followed by a validatory pull-down found that from about 90% of the human genome, they were able to identify and validate 50,000 direct protein-protein interactions. So these are direct interactions between partner proteins as opposed to protein interaction networks. If you consider that the human genome encodes about 23,000 protein encoding genes, only looking at the targets, the 23,000 putative targets, we're only looking to drug about 32% of the opportunities available to us. If we increase that into the protein-protein interaction space, we're giving ourselves a lot more opportunities. Now, this is, of course, a simplified demonstration, but I think that the sentiment holds quite firmly. Now, that's all well and good. However, the problem with protein-protein interactions is that they're quite featureless and flat. They don't have the molecular topography of a binding target. So these interactions don't really interact well with ligands which conform to our traditional notions of drug life. They also aren't associated with endogenous ligands such as ATP or NADPH, if the case may be. So using bioassay or developing bioassay is quite a challenge. So to sort out the bioassay problem, this is typically precipitate, this results in the development of these bait-prey interactions where you'll get either formation or disruption of a protein-protein interaction and subsequent fluorescence or fluorescence quenching in the presence of an inhibitor. This is, of course, has also been very successful. However, it does have its own drawbacks. It tended to have a high quantity of false positives, either from interactions with the fluorescence methodology, from planes type molecules, or its inability to actually detect genuine target binders at these quite flat interfaces. And it's this problem which is seen in the incorporation of biophysical methodologies such as typically NMR and crystallography incorporated to protein-protein interaction drug discovery to try and validate binding. So an important consideration is, of course, the classes of direct protein-protein interaction. So you get two classes in a general sense, transient and permanent, and transient is slightly weaker and permanent is much stronger. Permanent interactions have large interfacial interactions as opposed to smaller interfacial interactions of transient interactions. Permanent PPIs are also more associated with discontinuous conformational epitopes. So what that kind of means is when a protein-protein interaction comes together, it's kind of like a protein. If your protein is a fist and your hand is the other protein-protein interactions, your fingertips which make the interaction. So it's a series of amino acid interactions which aren't necessarily as a linear epitope, making this discontinuous interaction. Whereas the weaker transient interactions are more closely associated with continuous linear epitopes. So that's a single peptide region in its either in primary or secondary conformation, which is primarily responsible for the protein-protein interaction. So this includes disordered peptides or flexible regions of globular domains. So whilst permanent interactions are more associated with antigen-antibody interactions and large oligomeric complexes, which are transient interactions which are involved in cell signaling and regulation of protein activity. And it's this space here where, at least I feel, is the biggest opportunity for drugging protein-protein interaction. Now, of course, transient protein-protein interactions, by their very nature, are very tough to get together. They're not particularly stable. So biophysical methodologies such as crystallography or NMR make it challenging to view an entire protein-protein interaction. So there's a lucky shortcut available because of the property that the protein-protein interaction is mediated by a linear peptide. You can actually isolate that single peptide, and it will, when it interacts with the partner protein, will tend to go back to its conformation it would as part of the protein-protein interaction. And these protein-peptide interactions are used as a model for protein-protein interactions. They significantly simplify the model and allow you to look at what are the primary interacting residues that will mediate the protein-protein interaction. Now, of course, it makes it sound incredibly simple, but anyone who's worked with crystallography or even NMR will tell you that it's extremely challenging still to get crystals or to look at these and to grow proteins with labeled media, so you can do suitable NMR experiments, etc., etc. These techniques, NMR crystallography in particular, tend to also be quite heavy on your sample, so they use a lot of sample to try to get the data you need. So this is where we start getting interested in this technology, in this approach. So we're interested in native mass spectrometry. So what that is typical normal mass spectrometry where you can transfer something from a liquid phase into the gas phase and you look for a mass. The only thing we do differently is we tone it all down. Tone down the voltages, we tone down the temperatures, so you can spray a protein into the gas phase and it retains its native conformation. What that also means is you can take liquid phase information and transfer that into the gas phase, so things like protein-liquid interactions. So here's a native MS, and this is our charge state distribution. So it's in this case 11, 10, 9, 8. In the mass spec, we look for a mass over charge ratio. And we see an apoprotein, all in red at the 9 plus charge state. And then a very distinctive change in M over Z correlating to the mass of a single ligand, showing that we're seeing ligand binding. Now, it's important to note that this is gives you different kind of information to crystallography and NMR, but it's extremely quick. These are typically between 2 and 3 minutes, or maybe 4-minute long experiments, and there's no requirement for labeling. You don't need to optimize your conditions excessively. There's no cross-linking, there's no tethering, there's nothing that needs to happen. It's a mix and spray scenario. So it's a very rapid methodology. It also doesn't use nearly the same level of sample. So we typically use picomoles of protein to look for our data. So revisiting the HOP H390 PPR, as we said, we want to disrupt this protein-protein interaction, and fortuitously, as a transient interaction, it is mediated by this C-terminus MEEVD pentapeptide of HP90 interacting with HOP, which is shown in green, specifically the TPR2A domain of HOP. And like many protein-peptide interactions, we have crystal structure, and it shows the MEEV peptide interacting with this arginine and lysine-rich carboxyl clamp region of TPR2A. So we synthesized an acetylated MEEV analog, we expressed TPR2A, and we subjected them to native MS. So this is just the isolation of the 8 plus charge states, and what we can see is apoprotein beautifully, and MEEV bound at about a 75% ratio. And these were incubated at a 1:1 molar ratio. It's about 75%. So we considered this our protein-protein interaction model, just like the crystallography or NMR data would. We've seen protein-ligand interaction. We then rationally designed 2 peptides where we bioassociatively replaced one of the glutamic acids with the tetrazole, so peptide 2 and 3, and we did the same experiments, and we see our apoprotein and one of our tetrazole-containing peptides, in this case peptide 3, bound at a similar ratio to MEEV. We also synthesized a cyclic MEEV analog, which was found not to bind under our native MS conditions. The key experiment came when we co-incubated them all together. So we've got apoprotein, MEEVed, and MEEVed and our tetrazole-containing peptide all co-incubated at a 1:1 ratio. And in a 4-minute experiment, what we found was that we could see our tetrazole-containing peptide binding at a similar abundance to what it had before, but we saw quite a significant drop-off in the abundance of our protein-protein interaction model. And we interpreted this as disruption of our gas phase model. Analysis of the full charge state distribution and the relative abundances showed that in the MEEV bound at roughly 39 to 61% ratio, and in the presence of one of our tetrazole-containing peptides, we saw a nearly 50% fall off in the abundance of our PPR model, indicating again quite substantial gas phase disruption. In this instance, peptide 3 performed slightly better than peptide 2, but nothing too significant. The key experiment after that came when we incubated this, when we subjected this to a protein-protein interaction assay, and this is a domain-domain interaction between TPR2A and the HP90 C-terminus, and both peptides showed a dose-dependent inhibition of the protein-protein interaction, with peptide 3 performing quite a lot better than peptide 2. And importantly, peptide 4 was not active. The non-binding peptide was not active in this assay. So we wanted to then start trying to figure out any structural reason for this difference in activity. So we went to a technique called top-down mass spectrometry. So what this is, okay, I'm going to walk you through this process. So here's a charge state distribution of ApoTPR2A, and tuning a quadrupole, you can isolate one of those ions. The same for the bound species. This is a MEEV bound species, tuning a quadrupole, and you can isolate that. Then what you do is you subject this to ECD fragmentation, and you can fragment the backbone, get a series of peptides and a mass spectrum, and all of these masses, all of these peaks correspond to a very specific mass of your fragmented protein. And what's more is because we're doing this in native conditions, it will retain things like post-translational modifications, and in our instance, it will retain ligand binding tuned appropriately. So just to describe what we'll be looking for, this is an example of, you can see TPR2A, and this is its amino acid sequence. And ECD fragmentation very specifically fragments between the amine and the alpha carbons. We get C ions and Z ions. So if we have a fragmentation at that alanine, between the alanine and the valine, what we're actually looking for is not an amino acid by itself. What we're looking for is a peptide which corresponds to the mass of the fragment all the way to the N-terminus. And this is what we call a C ion. And then you get the corresponding Z ion, which corresponds to the mass of that cleavage all the way to the C-terminus. And then, of course, you get corresponding fragments. So piping this into our data, we're looking at the APO-TPR2A, and we see this fragmentation pattern. And the first thing to look at is in the N-terminus, we see a large number of fragmentations. And what this typically indicates is a little bit of instability in that region. So you get a lot more fragmentation in instances where you're losing some stability and you get fewer electrostatic interactions. Whereas in the C-terminus, you see a lot less fragmentation, indicating it's probably a bit more stable. When we piped in our MEVed, what we see, interestingly, is in our N-terminus, we see a lot less fragmentation, indicating that the MEVed, when it binds, and this is probably not that surprising, increases the stability of the system and you see less fragmentation. When we alter our search algorithms to look for fragments which contain MEVed, so that's the N and C terminus have been modified, we see a series of fragments where you can actually see MEVed binding. So we can confirm that MEVed is still binding when you fragment this protein. And just to analyze, we see a fragment like this, it might indicate that you're binding to that key lysine. However, what it actually means, this data means, is that it's binding somewhere in that region of the protein. So it's not too useful. When we did the corresponding experiment with our tetrazole containing peptides, what we find is that in the presence of the tetrazole peptide, we see our N-terminus becoming a lot less ordered. We see a lot more fragmentation, indicating that we're seeing a lot more disorder in this region. In fact, and we see a little bit more disorder in the C-terminus region as well. So this possibly gives us a reason for our activities, that the binding of the tetrazole containing peptide actually destabilizes the TPR2A domain and destabilizes formation of the TPR2A, but we're not too sure about that yet. Again, when we look at our, if we look for fragments which contain our tetrazole, key peptide, a key fragment we find is this one here on the aspartic acid. And what that means is that because it's a C ion going towards the N-terminus, it means that this entire alpha helix in yellow, or somewhere along this alpha helix, has our bound peptide. And this indicates a much more narrowed region of where you might see binding. Now, what's important about this is that these three lysines in a row, this triple K region here, lysine, 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 have been shown through mutational experiments to be critical secondary regions for protein-protein interaction. So if our peptide is interacting with one of those lysines, it might actually explain structurally why we are seeing this protein-protein interaction disruption more effectively than with MEVed. Okay, so we've done a little bit of SAR with these peptides, and we've danced the amino acids around, and this is just a subset of the compounds we've made, but the interesting ones I want to point out are these highlighted here. So we see a little bit of activity when we isosterically replace the glutamic acid with a phenylalanine containing analog. When we add a second tetrazole, we see similar activity to peptide 3, which had a single tetrazole in this region, and peptide 3. When we alter the stereochemistry of our position 3, so we make the epimer of our active peptide, we see no activity. So that's the MER-TRVD. And when we isosterically replace the glutamic acid, the terminal aspartic acid with tetrazole, we don't see much activity to write home about. So we were fairly satisfied with the series of results, but we thought, can we actually start looking for something more biologically relevant? So the first port of call was to look for full protein-protein interaction inhibition. So this is now full hop at about 60 kilodalton with full H390 at 90 kilodalton. And peptide 2, which had the tetrazole in the second position, was not really active, but METVD, as we call it, and N-double-TVD were both shown to inhibit the full PPI in a dose-dependent manner. And this we felt was a remarkable result, because these are two huge biomacromolecules coming together, or being prevented from coming together by a single 5-mer peptide, which is very targeted in where it binds. We put our peptides on a solid on a cephalose bead, and we were able to show that when incubated with a cell lysate, that's both the METVD and ME-double-TVD, were able to pull HOP out of the cell lysate, indicating that it does in fact interact with HOP in a biological context. And our control substance there's a little shadow of HOP coming out there, but it didn't really interact too strong, indicating our compounds are pretty good and strong interactors with HOP in a biological context. We then wanted to look at some more translational work, and to that end, we were interested in Kaposi sarcoma and herpes simplex virus. Now, KSHV, as it's called, is a class 1 carcinogen, and it's responsible for the formation of Kaposi sarcoma, which is a cancer which disproportionately affects the African continent, and it specifically targets immunocompromised patients. There's no targeted therapies, which is one of the major problems. But we became interested in this because of this part of the KSHV life cycle. Now, this virus goes into a latent phase where it kind of goes to sleep, and then can switch to its replication where it starts making new viral particles, and it's this part of the life cycle which is responsible for tumor formation. And it's this conversion from latency to its replication, which at least partly mediated by host HSP90. So we thought if we can inhibit the protein-protein interaction, perhaps we can prevent the onset of latent replication. So our peptides did nothing, but we then tethered it, possibly because of cell penetration, so we tethered it to an RH8 cell penetrating peptide, and then we started seeing some cool stuff. So to measure to detect whether we're inhibiting the conversion of latent to lytic replication, you start looking for transcriptional factors which are indicative of latent replication of KSHV. And these include things like OR57 and VRF3. And these are transcription factors for lytic KSHV replication. And in the presence of both our peptides, we see both of these markers are, the translation of both these markers is reduced, indicating that we are seeing less lytic replication because the current markers aren't present at least. We also saw this at a transcriptional level where some of the genes of important markers of lytic replication are also not being transcribed from the genome. So together, this indicated that these compounds, through their work, through their putative inhibition of the protein-protein interaction, are actually capable, at least from our preliminary evidence, of inhibiting the conversion of KSHV from latent to lytic replication. Okay, and of course they were shown to be, also importantly, they were not particularly cytotoxic, 79% cell viability, 25 micromolar. It's not what one would consider particularly cytotoxic. So we have what looks like non-cytotoxic inhibitors of Kaposi sarcoma, herpes simplex virus replication. So in summary for this, our native MS protein epitope model was shown to be a competent proxy of our protein-protein interaction, simple experimental setup, simultaneously confirmed genuine binders operating competitively. Forgot to tell you how that works. I can answer that as a question later. No assay interference, and has screening potential for more inhibitors later downstream. Also, these probe peptides demonstrate the potential for this PPI as a target for Kaposi sarcoma. Okay. Now, the application. So, of course, we've made this protein-protein interaction model. This could be used for screening, but a big drawback in the forefront of our mind is that we are still reliant on the presence of structural information to develop our protein-protein interaction model. So this hampers us for things like monitoring effective mutations. So what if we have an emerging variant where there's a mutation disrupting protein-protein interaction drugs or strengthening a protein-protein interaction? Can we respond to that without requiring preliminary or crystallography data or NMR data before we set up our experiments? And also, can we be responsive to new protein-protein interactions as they get discovered? Can we characterize unknown PPI without requiring structural data? So when building new protein-protein interaction models, it's actually quite a tough thing to determine what the binding epitope is. So even with crystallography, if you think about a protein-peptide interaction, how do you know what that peptide is? It's a tough thing to do. So some methods of epitope scanning include methods like PepScan. So what that is a systematic synthesis of a predetermined number of peptides of the whole protein interaction sequence. Okay, and then what you do is you incubate that with your partner protein and see what binds, or you see if it inhibits the protein-protein interaction, or whatever the case may be. However, of course, synthesizing that many peptides is incredibly expensive and incredibly labor-intensive. So it's not a particularly quick way or responsive way of responding to PPIs. This has largely been superseded by technologies such as phage display, and what this is a small phage is incubated with a custom DNA library, and it expresses a peptide on the surface of the phage, and you see which phages essentially bind to your partner protein. Now, it's extremely quick and extremely powerful. However, phage display only selects for binding. It doesn't necessarily represent partner protein. So the sequence will be the one that binds the most strong or the strongest, but it won't necessarily represent your partner protein, nor will it interact with the interface. So we thought, can we use some of the shortcuts provided by nature, but while still maintaining some of the kind of rigor of a approach like PepScan? So what we thought was, can we take a partner protein, digest it using some kind of trypsin or pepsin digestion methodology to get a very specific subset, predictable subset of peptides, which we can then incubate with our partner protein, other partner protein, do native mass spec, and start determining which peptides bind, and start from that elucidating what could be the likely location of the hotspot residues. And from that, use it to either build a protein-protein interaction model, or at the very least, use that information to feed into other programs which are still looking to identify the binding epitope. So we revisited the H3D TPR2A interaction because we were familiar with it, and we chose to digest it with trypsins. The reason for trypsin was trypsin digested lysines and arginines, which are highlighted in red and green, but specifically because there's an arginine just prior to the MEV region. So we thought this would be a nice way of determining, can we pick this up? And we also used solid phase supported trypsin, because what was important about that is we could do our digest and then immediately just remove it out of the crude mixture without purification, because the trypsin will stay behind, incubate it with our partner protein, and directly infuse it. So we digested up our H390 C terminus, and looking at this region of the mass spectrum, we found a series of peptides, and this is one peptide which doesn't contain MEVed, the same peptide with MEVed attached to it, and a series of peptides with and without MEVed, and we saw the same peptides throughout the mass spectrum. Okay, so we proved we can digest proteins. Very, very spectacular. Then the crux came when we co-incubated a TPR2A. So here's our charge state distribution of APO TPR2A, that's it in blue, and then we started seeing binding peptides. So that's that peptide bound, and we could determine what it was very specifically by the delta M over Z and the charge, and the isotopic distribution of the peaks as we saw them. So we could determine the charge state. What we found was all the MEV containing peptides that we'd seen were all able to bind to TPR2A. And in addition, we also picked up 2 peaks corresponding to MEVed itself. Now, we didn't pick this up in the crude mixture of peptides, but it in fact enriched when incubated with the protein. It's like the protein pulled it out of the solution. So we were able to determine, well, importantly, we didn't see binding from any peptides which didn't contain MEVed. So we were able to quite quickly determine that whilst we already knew MEVed was the binding epitope, we were only able to select for peptides which contain MEVed. So if we didn't have this information prior, we'd quite quickly be able to determine that it's a very likely hotspot region. Okay, so in summary, we were able to rapidly elucidate that epitope, and that was an hour digestion with the formative mass spectrum. We have the potential to apply this to uncharacterized PPIs. So we're looking quite heavily to find money to do this kind of project now. This will increase our access to this underexplored region of chemical space and drug design, augments current interfacial elucidation methods, and hopefully be able to rapidly respond to mutations as they emerge in the environment. Excuse me. And this will require only sequence data as opposed to full-scale structural data. All that leads me to do is to my acknowledgements, my very important collaborators, Professor Adrian Atkins, who is no stranger to this audience. We're very close collaborators, and she's an absolute world leader in the H390 space, or the chaperone space in general. Dr. Dave Clark from the University of Edinburgh. Also, he's my mass spec guru. We've been collaborating for a long time on our mass spec, and Fernando and Beatrice, who are peptide world leaders, and they've helped us a lot with peptide synthesis. Excuse me. And I just want to highlight Glenn Matendra, excuse me, who led the extracellular H390 product, and she's just graduated with a PhD, so congratulations to her. And then just to thank the very generous funders. Thank you very much.